Okay, students, welcome back. Today's lecture is going to be on chapter 13, just neural tissue. So this is your introductory lecture to the nervous system. After your second exam, we'll learn more about the details of the nervous system. So this is really just an overview on neural tissue. So we left off on muscles, and that muscles need to be told to contract by some sort of nervous stimulation. So that means that the nervous system has the ability to control and adjust the activities of other systems, not just the muscular system, but other body systems as well. Not only does the nervous system have the ability to control other systems, so does the endocrine system. And we don't have time to study about the endocrine system, but you will get lectures on that when you get to physiology. But just so you know, both of these systems have the ability to control other systems, or at least control the activities of other systems. So the nervous system usually, because it's communicating via action potentials or really just electrical signals, it has the ability to create really swift responses, but those responses don't tend to last very long. And that's in contrast to the endocrine system that communicates with other tissues, but the effects are seen much more slowly, and also they don't last as long. Okay, so here's your overview of the nervous system. Try not to let all the terminology intimidate you. This is really one of those systems that once you understand the terminology, it will be a whole lot more easy for you to understand the system as a whole, really. But if we just look at how the nervous system is broken down, it's broken down into two main subdivisions. And those two main subdivisions are the central nervous system and then the peripheral nervous system. And the central nervous system is pretty easy to understand in that it includes the brain and it includes the spinal cord. So really all of that neural tissue that's centrally located. And the functions of the central nervous system, or the brain and the spinal cord, are really to take in or integrate information, make sense of that information or process it, and then coordinate the appropriate response. So let me give you an example here. If you were to step on a nail, for instance, with your bare feet, there would be some information that travels from your foot all the way up to the spinal cord. And the spinal cord, the neural tissue in the spinal cord is going to integrate that information or really kind of make sense or take it in and figure out where that information needs to be sent. Does that information about you stepping on a nail need to go all the way up to the brain for higher processing? In some cases it does, and it depends on what the stimulus is. I'm just using a really easy example here. And then somewhere along the way, after that information, let me draw the stimulus here. So let's say you step on a nail, and that sensation of pain, it travels all the way up those nerves to the spinal cord. And in some cases, that information can be processed right there in the spinal cord. Like I said, totally depends on the stimulus. Sometimes information has to go all the way up to the big guy upstairs for processing. Regardless, that information is going to be processed. And what happens is then either the neural tissue in the spinal cord or the brain coordinate an appropriate response. And that appropriate response is going to be, in many cases, a signal sent down to a muscle. So again, let's go back to the example of you stepping on a nail. So that information is going to travel back down from the brain, back down the spinal cord, and then ultimately down into the muscles nearby where you stepped on that nail, and those muscles are going to contract. And they're going to contract in such a way to remove the stimulus. Or another way to say that is pick your foot up off of the nail. Now, this is a really, 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 really simple explanation of nervous system pathways, but it allows you to understand that the brain and the spinal cord, they take in that information from the peripheral nervous system, process it, and then coordinate the appropriate response to a stimulus. Now, more specifically within the brain, we've got a number of centers or a number of regions in the brain that are responsible for human intelligence. That's debatable. Memory, where we store certain memories of events or feelings. Certain centers in the brain allow for learning, and other centers allow for emotion. The second subdivision of the nervous system is the peripheral nervous system. And the peripheral nervous system is, or includes, all of the neural tissue that doesn't exist in the brain and the spinal cord. So all of these little yellow lines that you see here, all of those are nerves. And nerves are made up of neural tissue. So all of this, all of these little yellow lines, those are all part of the peripheral nervous system. So we say that the peripheral nervous system contains all the neural tissue outside the central nervous system. And I like to think of the peripheral nervous system as a two-lane highway. So let me go ahead and erase some of what I drew earlier. Give me a second here. And let's talk about these two pathways, or these two lanes on these two lanes on the highway of information, so to speak. So the peripheral nervous system is going to have two main pathways, a pathway where sensory information makes its way into the central nervous system, and motor information leaves. So let's go back to that example of you stepping on a nail. So one pathway is going to be a sensory pathway that's going to be carrying only sensory information. So sensory information might be like pain or changes in temperature or the feeling of something stretching. Those are all what we would consider sensations. So you could put your hand on a hot stove, and that information travels through the sensory pathways of the peripheral nervous system and sends that information to the brain and the spinal cord for processing. Now remember, once that information makes its way to the brain and or the spinal cord, that information is going to get sent up to the brain, and the brain is going to coordinate that appropriate response. And the appropriate response, in many cases, is going to be some sort of signal to a muscle to contract. So if you're talking about the stepping on that nail, that information is going to leave back out down to the muscles that are going to help you contract to move your foot in order to remove that painful stimulus. Or if you're talking about, for instance, putting your hand on the hot stove, motor information is going to leave out the peripheral nervous system, these pathways, the motor pathways, and make its way to the muscles that are going to help you to move your arm or move your hand away from that hot stove. So two pathways of information in the peripheral nervous system: sensory pathways and motor pathways. Let's look at it a little bit more closely. We can divide the peripheral nervous system into two subdivisions. And this is what I was referring to earlier as two highways or two lanes of a highway of information. There's the afferent division. Sometimes that's just called the sensory division. And the afferent division includes all of the neural tissue that is carrying sensory information. So we're carrying sensory information from the periphery back to the brain and the spinal cord. Remember, sensory information can be things like pain, changes in temperature, stretch, those types of things. So think about in your body where these types of sensations can be felt. Anywhere you can feel them or even not consciously feel them, those sensations make their way through the neural tissue of the afferent division of the peripheral nervous system, ultimately back to the spinal cord or the brain. So let's look at this. You've got sensory receptors called somatic sensory receptors embedded in your muscles, in your joints, and in the skin. So sensory information can come from your muscles, can come from your joints, and can come from the skin. Let's remind ourselves, for instance, of a Pacinian corpuscle. Remember, the Pacinian corpuscles are those sensory structures embedded in the skin that allow your brain to sense deep pressure or deep touch. Those Pacinian corpuscles send that information back to the spinal cord and or the brain for processing. Think of sensory receptors, pressure receptors you might have in your joints. Now, we didn't talk about this when we were learning about joints, but there are pressure receptors in there. These pressure receptors allow your brain to be consciously aware of any pressure you're putting on joints. So think about getting up out of bed. You immediately know or recognize that you're standing up rather than lying down because there's pressure now in your joints. Another place where sensory information comes from are the visceral sensory receptors. Remember, visceral or viscera means organ. And so you've got sensory receptors embedded in your internal organs that help your central nervous system monitor what's happening in your internal organs. So let's say the stomach as an internal organ, there are pressure receptors embedded in the muscular wall of the stomach that send signals to your brain when you're full. You could also think of another example that's very similar. You've got pressure receptors embedded in the wall of the bladder that allow your brain to recognize that the bladder is full. And then lastly, information is coming from the special senses, more specifically eye, nose, tongue, and ear. So whatever it is you're seeing, smelling, tasting, or hearing, that information gets sent through the afferent division of the peripheral nervous system back to the brain for processing. So all of these locations, skeletal muscle, joints, skin, internal organs, and then the special sense organs, they're sending information back through the afferent division of the peripheral nervous system to the brain and the spinal cord. And the brain and the spinal cord are going to take that in, integrate it, process it, and then coordinate the appropriate motor response. So motor information then is going to make its way out of the peripheral nervous system, out of the central nervous system via the efferent division, you can say efferent, or motor division of the peripheral nervous system. So here's the second lane of highway of information travel, where now motor information is leaving. So whereas the afferent division included all the neural tissue that's collecting and sending sensory information to the brain and the spinal cord, the efferent division is sending motor information from the brain and the spinal cord out to what we call an effector. And an effector can be either a muscle or a gland. Not all responses to a particular stimulus require a muscle contraction. Some stimuli require a gland to reduce some sort of hormone, or could be sweat or some sort of secretory product. So the efferent division of the peripheral nervous system is also called the motor division, sending information out to a muscle or a gland. So let's consider where motor information may go to, the different places in the body that motor information may go to. Well, motor information can travel out to skeletal muscle, and that would control skeletal muscle contractions. These aren't always voluntary, sometimes they're involuntary, for instance, like in a reflex. So blinking the eye or the startle reflex, those would be contractions of skeletal muscle, but they're involuntary. So that information travels out a special subdivision of the efferent division called the somatic nervous system. So the somatic division of the efferent division of the peripheral nervous system is sending out information to skeletal muscle. The second subdivision of the efferent division of the peripheral nervous system is what we call the autonomic division, or the autonomic nervous system. This division is sending out information, motor information, to your internal organs. Remember earlier we learned that there are sensory receptors embedded in your internal organs? Well, in order to respond to those sensory stimuli, the central nervous system will send out information through the efferent division down to your internal organs to respond to those stimuli. So this is going to regulate smooth muscle contraction. It's going to also regulate cardiac muscle contraction, not the actual beating of the heart itself. That is done through specialized cells. But the autonomic nervous system has the ability to control the rate at which your heart beats. And then also information can be sent out to glands to cause glands to release some sort of secretory product. All of this is involuntary. You don't have to think about smooth muscle contraction or cardiac contraction or glands secreting their product. It's really all automatic via the autonomic nervous system. Okay, so here's a flow chart given to you by your textbook, and you know how much I like concept mapping. It really helps you to visually process information all in one nice big picture. So let's just kind of wrap up the organization of the nervous system by looking at the components of this flow chart. So remember that the central nervous system is made of the brain and the spinal cord, and that's where information processing occurs. Information that the brain and the spinal cord are processing comes from your internal organs, can come from skeletal muscle, joints, or the skin, but also comes in through special sensory receptors. So all that information is collected and sent up to the brain via the spinal cord for processing. Now the brain is going to respond to those stimuli and send out information via the efferent division of the peripheral nervous system. So depending on where that information has come from, motor contraction or motor commands are going to go out to skeletal muscle, or motor commands can go out via the autonomic nervous system right out to smooth muscle, cardiac muscle, and glands. All of that is involuntary. Let me erase that. So those commands are involuntary. These commands going out the somatic nervous system, those are all voluntary. So you really have areas here where you're collecting information, some sort of stimulus makes its way up to the brain, and the brain responds to that stimulus with some sort of motor command. And depending on where that motor command needs to go, it will leave out either the somatic nervous system or the autonomic nervous system, and ultimately end up either at skeletal muscle or smooth muscle, cardiac muscle, and glands. So I encourage you to draw your own concept maps while you're learning this, just so that you get really good at the terminology and understanding these pathways. Okay, so that's the organization of the nervous system. Now let's move into understanding neural tissue. And neural tissue is the fourth and final tissue in the human body that we're going to be learning about. So let's kind of think about the hierarchy of organization, how organisms are put together here. You know that cells come together to make tissues. Tissues come together to make organs. And organs come together to make organ systems. So we're going to be learning about neural tissue. We are right here in the levels of organization, and we're going to learn about the cells that make up nervous tissue. So there are two main types of cells. What we call nerve cells, another name for those nerve cells would be neurons, and there are neuroglia cells. And there's a very distinct difference between these two types of cells in that neurons have the ability to generate an action potential, or this electrical signal. It makes them really special, makes them very unique. This is how they're able to process information and transfer information from one neuron to the next, or, as we learned in the muscle lecture, from one neuron to a muscle. So they have this really special ability to generate an action potential. Whereas neuroglial cells are really just supporting cells. They're there to support and protect neurons. You can kind of think of neurons as being, let me get back to a different color here, neurons as being kings and queens, in that they're very unique. They are lesser in number than neuroglia. You could think of neuroglia as being subjects in your fictitious kingdom, where the subjects are much more numerous and not as important as the kings and queens. Kings and queens in your fictitious kingdom, they're going to have the ability to generate, I'll just abbreviate it, generate an action potential, where neuroglial cells don't. They're there to simply support the kings and queens. So neuroglia, as the subjects, they're going to be there to protect the neurons. Neuroglia do not have the ability to generate an action potential. So those are two big differences between them. Another big difference is that neurons cannot carry out mitosis. So they do not undergo mitosis. They cannot grow and divide once you pass a certain age of development. So they do not undergo mitosis. So when they're damaged or when they die, they cannot be replaced. Whereas neuroglial cells, they do undergo mitosis. They do, when they get damaged or they are about to die, they will undergo mitosis. They'll make a copy of themselves. So putting this into context, neurons are nerve cells, with the one exception of a childhood cancer. Nerve cells do not create brain tumors. The cells responsible for creating brain tumors or becoming cancerous are neuroglial cells. Remember that cancer is uncontrolled mitosis or uncontrolled cell growth. So Neuroglial cells, they undergo mitosis, so they're going to be the cells that can become malfunctional and undergo mitosis very rapidly and create brain tumors. Whereas neurons, they do not undergo mitosis, so they don't have the ability to grow and divide out of control and become tumors. Let me give you an example of a specific type of brain cancer. There's a type called astrocytoma. You'll learn here in a few minutes that an astrocyte is the name of a glial cell. So an astrocytoma is a type of brain tumor. Okay, so let's look at the structure or the anatomy of just a really generic neuron. In a minute, I'm going to show you that neurons can come in different shapes, different structures. But let's just understand, just in general, the structures here. So neurons are going to be cells that are going to transmit information in one direction only. So if you think about it, oops, let me change that. That is not a good color. Okay, hang on. Let's do blue. So neurons are going to have, just in general, a receiving end for information and a transmitting region, and then a sending region. And what I mean by that is these little structures that you see here, these little branches, these are called dendrites, and they're going to receive some sort of information, some sort of stimulus. That could be a chemical stimulus, meaning that could be some sort of molecule that binds to the dendrite, or it could be some sort of physical stimulus, physical compression or some physical distortion of those dendrites. So that's where information gets received on a neuron. And then the region of a neuron that actually processes it, kind of like a little microchip or a microprocessor, is this structure, the cell body. That's where you find the majority of the organelles, like that structure right there. That would be the nucleus. So that's the processing center. Leaving the processing center, or going off to the right here, this long structure, that's called an axon. That's the transmitting structure. The way in which neurons work is that as a result of some sort of stimulus received here, the cell body is going to generate an electrical signal. And that electrical signal is going to travel out through that axon all the way out to the ends, the little tips of these fine branches down here. So in that respect, the axon is a transmitting structure. It's transmitting an electrical signal. So let me go ahead and erase some of this. So that axon then transmits that electrical signal, also known as an action potential, transmits that signal down to or through, let's just draw the action potential again. It's going to send that information out through these structures. Now those little structures in the end, they're not part of the axon. They're what we call telodendria. Telodendrian would be singular. Telodendria is plural. Right here at the end are these little bulbous structures, and those are called synaptic boutons. And in those synaptic boutons, let me see if I can find a good color here, are little packages of molecules that we call neurotransmitters. So when that electrical signal reaches the synaptic boutons, it actually causes those neurotransmitters to leave the synaptic boutons and end up in this little space right here. And so it's these synaptic boutons that send chemical information to an awaiting cell. Well, the awaiting cell could be another neuron, so here would be the cell body with its dendrites. And those chemicals, they may bind to these dendrites here. And then that next neuron receives that information. Let me just draw one here real quick. That next neuron receives that information in the form of a chemical, and then that next neuron then here in the cell body, that cell body processes that information and generates another action potential that travels out an axon, travels out the telodendria, and then ultimately is going to cause chemicals to be released from the next synaptic boutons. So you can see how if you line up neurons end to end, they create these pathways of communication. This is exactly what happens with muscle cells. A motor neuron cell body generates an action potential. That action potential travels down the axon and ultimately ends up right here and releases molecules, little molecules of neurotransmitter that bind to the muscle cell, and that causes the muscle cell to contract. Now, under normal circumstances or in a normal semester, we would have learned about all the intermediate steps which cause muscle contraction, but I've eliminated that. So if a lot of this doesn't make sense to you as far as muscle contraction, that's okay. The important thing to understand is the anatomy of a neuron, where information is received here, that the cell body processes that information and generates an action potential. The axon sends the action potential or transmits the action potential, and the synaptic boutons are responsible for releasing chemicals, in this case neurotransmitters, that will cause or will create communication with an adjacent cell. Could be another nerve cell, could be a muscle cell. All right, on the last slide, I said you were looking at the shape of kind of a generic neuron. And it's actually not really a generic neuron. It does have a name, which I'll show you in a few more slides. But when I say generic, I mean it's really the one that students first learn about the function of a neuron on. In a psychology class or maybe in a physiology class, you've probably learned what a neuron looks like by looking at a very similar picture. But they don't all have that shape. They can vary in their shape. So on this screen you see what we call a bipolar neuron and a pseudo-unipolar neuron. Now all neurons function generally in the same way in that they receive some sort of stimulus, that stimulus is processed, and then the cell body generates an electrical signal, an action potential. And that's still true with these two shapes of neurons, except now the type of information that is being processed and transmitted is different. The structure is also different. So if we look at this neuron right here, look at how much shorter it is than the neuron over on the right. So structurally, they're a little different in their length, and also they're structurally different with regards to the cell body. So the cell body in a bipolar neuron has two branches coming off the cell body right there. So that's why this neuron gets its name. Whereas a pseudo-unipolar neuron or unipolar neuron only has one branch. Look how much longer that pseudo-unipolar neuron is than that bipolar neuron. And look at the number of branches coming off the cell body. Those are the structural differences. Now let me erase some of this. And let's talk about the functional differences. So functional differences between the bipolar and pseudo-unipolar are that the bipolar neurons, they're only transmitting special sensory information. So they're going to carry information from the eyes, the nose, the mouth, and the ears back to the brain. They're sensory neurons, but they're only special sensory neurons, meaning they're really highly specialized. Whereas pseudo-unipolar neurons, they're transmitting some general sensory information about pain or changes in temperature or stretch. These neurons are found only in the peripheral nervous system. Only in the peripheral nervous system. They're carrying sensory information from the periphery back to the central nervous system, the brain and the spinal cord. Okay, here's the neuron shape that we were first learning about a neuron on. And what you saw earlier is what we call a motor neuron, also known as a multipolar neuron. Multipolar refers to the number of branches coming off the cell body. So it's got many branches. It's also called a motor neuron because functionally it's sending motor information to a muscle. And motor means movement. Now look at the structural difference here. Not only does it have many branches and the cell body is located just at one end, look at the overall length of a multipolar neuron. Some of them can be over a meter long. That's 3.3 feet. Location-wise, let me change colors here. The cell bodies and the dendrites are located in the central nervous system. And the axon, the telodendria, and the synaptic boutons are located in the peripheral nervous system. We'll learn more about that when we get to the spinal cord. Okay, so on the previous slides, we looked at how neurons are classified structurally. What's their shape? Those same neurons can be classified functionally based on what they do. So this is another classification system that we can use to refer to neurons. So there are sensory neurons. All right, so sensory refers to function. Unipolar neurons, or, let's see, I didn't write it here. Unipolar or bipolar neurons, those are sensory neurons. So most of their axons, dendrites, and cell bodies are in the peripheral nervous system. So most of their axons, dendrites, let me just do this here, and cell bodies are located in the peripheral nervous system. There are some exceptions to this, but this is generally where those neurons are located. A motor neuron refers to a neuron that's causing some sort of motor movement. So motor neurons are multipolar in shape. As I mentioned earlier, their dendrites and their cell bodies are the central nervous system, and their axons are found in the peripheral nervous system. Their axons, or more specifically, the telodendria and synaptic boutons, communicate or synapse with what we call an effector. An effector is a muscle or gland. And then lastly are what we call interneurons or association neurons. Most of them are multipolar in shape. So in this case now, multipolar neurons aren't used to cause some sort of movement. These multipolar neurons are acting like middlemen or middle people. They're processors in between. So they're not causing any type of movement. But what they're going to do is integrate information that comes from sensory neurons and send that information or route that information to the appropriate motor neurons. So sensory information comes in from the periphery. It first might be received by an interneuron or an association neuron, and then that association neuron sends that information directly to a motor neuron. So let's do a pathway here. Sensory neuron sends sensory information to the central nervous system, and in some cases, that sensory information is received by an association neuron. And then that association neuron can send that information directly on to a motor neuron. Again, we'll learn more about this when we get to the spinal cord. Okay, we just finished up understanding the shapes and functions of nerve cells, neurons, one type of cell found in the nervous system. Now we're going to move on to the second type of cell found in the nervous system called neuroglia. And remember that neuroglia do not conduct electrical signals, do not conduct action potentials. They're there to support neurons. So if you go back to the analogy I gave you, neurons being kings and queens and very special, not having the ability to undergo mitosis, you have the neuroglial cells, the support cells that are supporting those neurons in any way possible, because those neurons are so unique, and if they get damaged, they can't replace themselves. So neuroglia play a really important role in the nervous system. In general, like I've mentioned, they're going to physically support neurons, or they're going to provide a framework or an internal skeleton, so to speak, for the neurons to anchor to. They're going to perform maintenance in the area surrounding the neuron, and in some cases they act as phagocytes, little Roombas that can clean up the area around neurons. And let me explain this a little bit more to you. Neurons are very finicky. They require a pristine and perfect environment surrounding them. So let's say that this is a neuron cell body. Here are the dendrites. Here's the axon, the telodendria with the synaptic boutons. The area surrounding a neuron has got to be perfect. This area is going to be filled with what we call interstitial fluid. That just really means the fluid in between. It's got to have an appropriate concentration of ions like sodium, calcium, magnesium. It's got to have the appropriate amount of water. It can't have too many waste products in it, waste products that have come from the neuron itself or nearby nerve cells. When this interstitial fluid or that environment outside of a neuron is not perfect or pristine or clean, the neuron will just malfunction. In some cases, the neuron will die. And you know that neurons can't undergo mitosis. So what some of these glial cells will do is they will keep all this area surrounding the neuron nice and clean, regulate the nutrients and waste products and ions so that that neuron can function properly. If you just look at the ratio of neurons to neuroglial cells, there are about 100 billion neuroglial cells for that make up the nervous system, and that's about 5 times the number of neurons that you have. So these glial sample here real quick. So imagine, or maybe you don't have to imagine, maybe this was last night, but let's say, for instance, you don't get a very good night's sleep, and the next day you're a little bit foggy, you're not thinking clearly. The reason for that, we think, is that when you sleep, let me do this with my eraser, when you sleep, there's a fluid that passes between neurons that cleans up this entire area. So this whole area surrounding the neuron gets washed away of any metabolic byproducts, any potentially high concentrations of molecules in the area that might be toxic to the neuron. That all gets cleaned up overnight so that the next day the neuron can work properly. But if you don't sleep well or haven't slept, that area doesn't get cleaned, that interstitial fluid doesn't get cleaned up properly. And so the next day, your neurons aren't functioning properly. A really important role. Let me give you... Okay, so let's look at the different types of neuroglia and where they're found. First of all, let's look at the central nervous system. There are 4 types of glial cells there. And then in the peripheral nervous system, there are only 2. Again, here's a nice flow chart to help you begin learning how these glial cells are organized. Astrocytes. These are one type of glial cell in the central nervous system. They're actually the largest in size, and they're the most numerous. They provide a number of valuable services for the neuron itself. Let's look down at the picture here. So here's a capillary. That's a really small blood vessel. Here's the astrocyte, so it's pretty small in comparison to the neuron. And here's the neuron itself. One of the things you can clearly see the glial cell or the astrocyte doing is with these little extensions right here, it's supporting that neuron. It's physically supporting or holding up parts of that neuron. Another thing it's doing with those extensions is holding the neuron here away from the blood vessel. That blood vessel cannot come any closer to the neuron because that astrocyte is physically holding it at arm's length away from the neuron. Well, that's good. This is what we call the blood-brain barrier. These astrocytes help to maintain the blood-brain barrier by physically keeping blood vessels away from those neurons. Let me tell you why this is important. In other areas of the body, blood vessels come very close to other cells. And under normal circumstances, let me use green here, under normal circumstances, in any other area but the brain, you might have ions freely leaking out of these blood vessels, and then those ions would diffuse to the necessary cells. However, because these neurons are so picky about their external environment, we can't allow, or the body can't allow, those things to freely pass out of those blood vessels. They need to be kept physically away from those neurons. So this astrocyte right here is going to regulate the space around it and physically keep that capillary away from the neuron. Let me erase this here. That astrocyte is going to also regulate the amount of certain nutrients that make it their way to the neuron as well. So one of those really important nutrients is glucose. Lactate is utilized by the neuron as well. That astrocyte is going to regulate the amount of those things that make their way to the neuron. So, that astrocyte regulates the chemical composition of that interstitial fluid. It helps to maintain the blood-brain barrier. It can also isolate neurons from one another. Neurons can't lie right on top of one another in order to communicate effectively. They have to be a very, very, very short distance away from one another in order to function properly. As you can see, also that astrocyte is providing a physical framework. And then in addition to that, that astrocyte can perform repairs. So if there's slight damage to like the plasma membrane of this neuron right here, that astrocyte can perform on-site repairs, minor repairs. Okay, the other three neuroglial cells that we find in the central nervous system are oligodendrocytes, microglia, and ependymal cells. So let's look at oligodendrocytes first. Oligo means several, and dendra means branches. So I kind of think of these as octopus glial cells, and like here would be the head of the octopus, and each one of these extensions are the extensions of the oligodendrocyte. Those would be like the tentacles on an octopus. These oligodendrocytes make a lipid called myelin, and they concentrate that myelin in the tentacles or in the extensions. And what they're able to do is wrap their extensions, wrap their tentacles around nearby axons. So this would be an axon right here. Here would be another axon of another neuron. Here's an axon of another neuron. And what you see is this oligodendrocyte kind of wrapping its extensions, its tentacles, around neurons. And that helps to insulate the axons of neighboring neurons. And when I say insulate, I don't mean that neurons get cold. I mean that because those axons are generating an electrical signal, transmitting an electrical signal down the length of them, we want to make sure that that electrical signal gets to where it needs to go. We don't want that electrical signal to get lost along the way. So the myelin in the oligodendrocytes insulates the axon, just kind of like the covering or the rubber coating on your cords to your devices. The electricity comes from the outlet, but you want the electricity to get to the light bulb. You don't want the electricity to get lost along the way. So we cover or coat our electrical wires so that the electricity goes straight from the outlet to the light bulb and doesn't get lost along the way and end up shocking you. Microglia are just kind of like the name implies. Microglia are small glial cells that are very similar to white blood cells, in that they're going to digest pathogens that maybe make their way around neural tissue and any cellular debris. By cellular debris, I mean any neighboring glial cells that may have died or to become damaged. The microglial cells are going to clean up that cellular debris, or any pathogens. Some pathogens are able to make it through the blood-brain barrier, and those microglia are going to quickly digest or phagocytize those pathogens. And then lastly, there are ependymal cells. Ependymal cells are going to be found lining the cavities of the brain and the spinal cord. You might think that the brain is a solid structure, but in fact, there are small cavities on the inside. Those small cavities make cerebral spinal fluid. There's another small cavity in the spinal cord where you find cerebral spinal fluid. So all of those cavities are lined with ependymal cells. So if we go over here, these are the ependymal cells. And you can see cilia on the surface. Not all ependymal cells will have cilia, some will have microvilli. But those ependymal cells are going to be found either lining the cavities in the brain or the spinal cord. They can either have microvilli or cilia on their surface, depending on their location. And what they're going to do is monitor the composition of what we call CSF, or cerebral spinal fluid. That's a fluid that you find within the brain, surrounding the brain, within the spinal cord, and surrounding the spinal cord. We'll learn more about that in the next few lectures. Okay, now let's move on to neuroglia of the peripheral nervous system. There are only two, so we've only got 2 slides left. One of the glial cells in the peripheral nervous system is called a Schwann cell, and you'll learn a whole lot more about Schwann cells when you get to physiology. But Schwann cells are these little orange cells or beigeish cells that you see here. And they're kind of like oligodendrocytes in the central nervous system, where they produce this lipid called myelin. But the way that they myelinate or insulate an axon is a little bit different. One Schwann cell can only myelinate a portion of an axon. So let's look at this one right here. So here's one Schwann cell just myelinating or insulating this one axon that's drawn in blue. So in the peripheral nervous system, you might have the axon of a motor neuron myelinated by several Schwann cells. There's one here, all the way down. But you see that there are spaces between those Schwann cells. Let me erase what I've done here so we can see those spaces. And those spaces are referred to as nodes of Ranvier. Where's that word? Here's that word, nodes of Ranvier. And these nodes of Ranvier are where the axon is not insulated. It's not myelinated. And that axon then allows for exchange of ions between the extracellular fluid and the intracellular fluid. Extracellular fluid meaning the fluid outside the axon, and intracellular fluid meaning the fluid inside the axon. So this is where a really important exchange of ions occurs. When you get to physiology, you'll learn more about that. And then lastly, there are satellite cells. This is the second type of glial cell in the peripheral nervous system. And what satellite cells do is they surround neuron cell bodies, and they're going to aid in regulating nutrients to neurons of the peripheral nervous system. So if we go down to the picture at the bottom here, here is let me get to the right pencil. Here is a neuron right here. And all of those little dots that you see surrounding that neuron, all of those would be satellite cells. Okay, that's the end of the Intro to Nervous System lecture.